The sampling site is located in Old Age Daycare Home of Badegaun, within Ward number 14 of Godavari municipality. The site is situated about 6 to 7 kilometers southeast of the Patan Industrial Estate along the Godavari Road. The containment is in the building premises constructed within the ground level. However, this placement leaves it exposed to direct surface runoff, and during rainfall, water can easily enter the tank. Access to the tank can only be gained by sliding the cemented slab that normally covers the blank. The researcher begins by arranging all the equipment and materials needed for sampling, such as core sampler, sample containers, measuring tape, permanent marker, ice box, tissue paper, measuring cylinder, bucket, and disinfectant spray. Before getting started, researcher puts on personal protective equipment such as a waterproof apron, gloves, rubber boots, masks, protective goggles. This ensures safety against harmful gases and pathogens. After this, researcher prepares the core sampler for the sample collection. For this, a metallic rod with an airtight seal at the bottom and a rope at the top is fitted inside a PVC pipe with a diameter of 50 millimeters. The tank stores black wastewater generated from 4 members residing in this institute. The tank was constructed in 2019. It is lined structure with semi-permeable side walls while the base is laid with brick works. The tank measures 2 meter 90 centimeter in length and 2 meter in width and has a depth of 5.5 feet. To determine its depth, researcher carefully lowers a long stick into the containment and then withdraws. The sludge leaves a clear mark on the stick, which is then measured with a measuring tape. The distance from the base of the stick to the mark is found to be 10 cm, which is the depth of the sludge. A sample is collected from this level to ensure it is truly representative. Researcher holds the core sampler device and lowers it through the opening of the tank to the bottom of the tank. After letting it settle for about 30 seconds, the sampler is sealed and removed. The collected sludge is then transferred into a bucket. This process is repeated until the bucket is full. Once enough sludge is collected, the researcher homogenizes the sample by mixing it thoroughly. The sludge is transferred into a 10-liter container and a portion of the sample is poured into a 1-liter transparent measuring cylinder and allow it to settle. The researcher then repeats the sampling process to collect the next 10 liters of fecal sludge. Type of fecal sludge is also determined by calculating the volume of the fecal sludge settled in the measuring cylinder. In this case, fecal sludge type is semi-solid and black in color. Finally, once all samples are collected, the researcher carefully cleans and disinfects all the equipment, ensuring everything is ready for safe storage and transport. After collecting the samples, the icebox along with other necessary materials are loaded into the vehicle for transport to the ENPHO laboratory. The lab is approximately 8.2 km from the sampling site, and it takes around 45 minutes to reach the lab. Upon reaching the ENPHO lab, researcher removes the sample containers from the icebox. The sludge is transferred into a 20-liter bucket and mixed thoroughly using a stainless steel ladle to ensure homogeneity. The mixed sample is then divided into 3 parts or duplicates. For the storage of the sample, researcher arranges the different volume of sample bottle based on different type of analysis. For the physiochemical parameters such as total solid, pH, TSS, turbidity, pH, EC, ammonia, sludge volume index, orthophosphate, nitrite. Researchers store the sample in 1-liter bottles without preservatives. pH and conductivity are analyzed immediately, while the remaining parameters are tested later after refrigeration. For the parameters such as COD, TKN, and total phosphorus, samples are preserved with sulfuric acid to keep the pH below 2. And for microbial analysis, researchers store the source sample in the sterilized containers. Throughout the entire process, standard sampling protocols are strictly followed, with careful handling at every stage to ensure accuracy and reliability of the results. This completes the process of fecal loss sampling, transport, and storage for laboratory analysis.